Welcome back. Last time, we discussed how the science of polymer chemistry was unlocked by the steadfast insistence of Hermann Staudinger and a few happy accidents by scientists and entrepreneurs in the late 1800s. But recall that the accomplishments of the Mayans, Charles Goodyear, and other early polymer chemists were all based upon the modification of naturally occurring polymers like polyisoprene in rubber or cellulose from plant walls. These polymers were not made by man, but were produced as part of the machinery of life. Now that we have a basic understanding of polymer structure and properties, we're ready to turn our attention to nature and appreciate what amazing polymer chemistry is going on around us, and even inside of us. When we turn our attention to the chemistry of life, we see polymers everywhere, from the material making up our skin and hair, to the molecules used to store essential energy needed for life, to the genetic code that defines us. Polymers are everywhere in living systems, and 3 billion years of evolution has led to some truly remarkable polymer chemistry. When we discuss polymers used in living systems, we often refer to them as biopolymers for obvious reasons. They're not terribly different from the semi-synthetic and synthetic polymers that we studied last time. Biopolymers are constructed from monomers, and they're often formed using condensation reactions, just like the one we use to make nylon 6,6. And they exist some as homopolymers and some as copolymers as well. But aside from having biological functions, what really makes biopolymers unique and fascinating is their complexity. Living systems exercise control over synthesis of these polymers that no chemist could ever hope to achieve in a beaker. And that makes them truly special. There are 3 broad classes of biopolymers that I want to discuss with you today. Those classes are polysaccharides, polynucleotides, and polyamides. Polysaccharides, as the name implies, are built from sugar monomers. We already looked at one example of this in our last lecture. Cellulose is actually a biological polymer, and the one that was characterized to finally settle the debate. But cells have devised even more ways to use polysaccharides as an energy storage system. Polynucleotides are formed using molecules known as nucleic acids. The classic examples of these are DNA and RNA. These polymers drive the storage and transmission of our genetic code. Finally, we have polyamides. Biological polyamides use the same linkage that holds nylon polymers together, but their complexity gives them vast functionality, from the collagen in your skin to the hemoglobin carrying oxygen through your body. Now, during this lecture, we'll examine all 3 of these classes of biopolymers and try to garner an appreciation for just how remarkable their structure and function are. Let's get started. Sugar. We're all familiar with it. From soft drinks to candy, there never seems to be a time when a product containing sugar is very far away at all. Chemists refer to sugars as saccharides. One of the simplest saccharides of all is this, glucose. Glucose is everywhere. One molecule of glucose can pair with another simple saccharide, fructose, to produce the compound that most of us know as table sugar, or sucrose. Sucrose is formed when a condensation reaction links these 2 units together in a very specific arrangement, creating a new compound with chemical properties slightly different than either of the individual components. This is also true of the sugar maltose, which forms when 2 glucose molecules come together to form what we would call a dimer, a molecule consisting of 2 linked units. These relatively small molecules can be very quickly broken down into glucose, which is burned for energy in your cells. But what happens when you have more glucose than you need? 3 billion years of evolution have tuned the cells in your bodies to know better than to be wasteful. When there's excess glucose available, the body does what any conscientious person would do with spare cash, food, or practically any other resource. It stores it for a rainy day. In your body, the storage system that your cells use to keep plenty of glucose on hand is a polymer. But the polymer that your body makes out of glucose is not the same as the cellulose from plant cell walls. Cellulose is too strong, and humans lack the ability to break it down to recover the glucose monomers inside. Instead, we use this polymer, glycogen. Notice the difference? Glycogen uses glucose monomers linked together in a different geometric arrangement than cellulose. The long, slender fibers of cellulose are replaced with a highly branched system of connected monomers. This different geometry changes the properties of the polymer, making it less fibrous and easier to break down chemically for quick energy use. But the versatility of polysaccharides doesn't end there. In an interesting twist, nature itself has created some semi-synthetic polymers. Molecules like the chitin that makes up the armor of arthropods like crabs and insects as well. The molecule chitin is actually a polysaccharide containing a chemically modified glucose molecule called N-acetylglucosamine. Now here's a small snippet of a cellulose polymer. Now let's look at chitin. Notice the new functional group? Such a small change to such a complex structure, isn't it? But it makes a world of difference. These new atoms change the way that individual strands of chitin interact with one another. And what was once fibrous cellulose is now a sheet of chitin armor. When combined with calcium carbonate salts, as it is in crabs, for example, chitin armor is so rigid and tough that crabs have to periodically evacuate their old shells so that they can grow. If you've ever wondered what chitin tastes like, just pay attention to the next time you're enjoying a soft-shell crab. Soft-shell crabs are trapped just after molting before their chitin shell has a chance to gather the minerals from the water it needs to become rock solid. But polysaccharides are not the end of the story on the polymers in life. Not by a long shot. As a structural material and for energy storage, polysaccharides are top-notch. They typically consist of just one or two types of sugar monomers, limiting the variability of their composition. Your glycogen and mine are exactly the same. But the body has another polymer at its disposal, one with tremendous variability, one that has so many possible combinations of monomers that each and every living thing on this planet has a different version of it, unless, of course, you're an identical twin. I'm talking about DNA. Deoxyribonucleic acid, a biopolymer so large and complex that it can store within it all the information needed to make you, you. The story of DNA begins in 1869, when Friedrich Miescher extracted this substance from the nuclei of cells that he was studying. Miescher dubbed this material nuclein, an indication of its origins. It was a Russian researcher by the name of Phoebus Levine who started scientists on the long road to understanding the structure of this critical material that Miescher had discovered. Levine was able to break the so-called nuclein down into its constituent parts. He found phosphates, 4 organic bases, adenine, thymine, cytosine, and guanine, and interestingly, a sugar called ribose. In 1940, nearly 70 years after Miescher's discovery of nuclein, Erwin Chargaff made a telling observation. He noticed that when he isolated all 4 of the bases found by Levine, cytosine and guanine were always present in equal amounts, as were adenine and thymine. It was an intriguing observation, and one that hinted at the structure of DNA. Tantalizing as this observation was, debate still raged about exactly what DNA was and how the molecule was put together. The world would have to wait another decade to settle this argument. The key piece of data that unlocked the secrets of this biopolymer was one from the same technique that had proven Hermann Staudinger right about cellulose, X-ray crystallography. The lab of Maurice Wilkins and Rosalind Franklin at King's College in London, as well as that of James Watson and Francis Crick, generated that X-ray crystallography data almost concurrently. A race to press ensued, and Watson and Crick prevailed, securing their place in history as the widely credited discoverers of this, the DNA double helix. Watson and Crick's structure revealed not only the elegant twist in this polymer of life, but its inner workings as well. Everything that was already known to be part of DNA was there. ribose-like units held together by phosphoester bonds. And attached to those ribose-like units were the 4 base pairs: adenine, cytosine, guanine, and thymine. The structure demonstrated why Chargaff had noticed the equal proportions of cytosine and guanine, as well as those of adenine and thymine. The solved structure of DNA showed 2 strands with complementary pairs of bases that came together, almost like the teeth of a zipper. Watson and Crick's structure allowed us to finally understand Chargaff's observation that G and C were always present in the same proportions, as were A and T in different DNA samples. So to demonstrate exactly why that is, I've shown you here these base pairs all put together in what are known as Watson-Crick pairs. Now, they're called Watson-Crick pairs, of course, because it was their crystal structure that allowed us to finally observe this. You'll notice here that A and T come together in such a way that a network of hydrogen bonds, 2 in particular here, hold them together in such a way that they hold the DNA, which is right here and right here. These are the connection points to the rest of the DNA molecule. They hold them together in such a way that that double helix can form and be held together tightly. If we look at G and C, you'll notice there's actually a network of 3 hydrogen bonds that can form between them, and that holds the DNA together even more tightly. But when we try to combine non-complementary pairs, if you will, like for example, let's take a G and a T and see if we can make them pair together. Well, when I put them in the orientation that they have to have in order for them to be in a DNA molecule, we notice something's wrong, right? Here we have an NH pointed at an NH, both of them trying to donate hydrogen bonds. This won't work out. Here we have a carbonyl pointed at a carbonyl. Again, these two they're primed to receive a hydrogen bond, but incapable of donating one. So that hydrogen bonding network that we would anticipate that would hold the DNA together simply isn't there when we try to pair G with T. Let's try to pair C with A. When we do that, we see the same kind of problem happening here. We have 2 hydrogen bond donors aligned, 2 hydrogen bond acceptors aligned, and down here we have a hydrogen that's not connected to the appropriate atom. So again, a network of hydrogen bonds holding together a piece of DNA with this kind of pairing simply doesn't exist. So it's the Watson-Crick base pairing that really drives the formation of DNA. As elegant and remarkable as all of these features of DNA are, what really makes this molecule of life useful is the fact that it is a polymer. DNA strands can reach truly staggering lengths in higher organisms, and you are a perfect example. The snippets of DNA that I've been showing you pale in comparison to the size of your actual DNA sequence. The human genome, as it's called, consists of some 5 billion base pairs strung together. That's enough DNA to create a 1.8 meter strand when extended completely, nearly as tall as I am. This makes DNA the undisputed king of information storage. 5 billion letters go into the code that defines who you are, at least in a biochemical sense. But DNA isn't the only polynucleotide that your body relies on. DNA is the vessel for genetic codes. It's the template, the ultimate source of information. So, like any archive, it's designed to last you your entire life. But the genes in your DNA are merely instructions. Those instructions have to be carried out. And the task of turning the information in your DNA into something else falls to a very close chemical cousin, RNA. RNA, or ribonucleic acid, is remarkably similar to DNA itself, but with 2 very special differences. The ribose sugar is not missing this hydroxyl group like DNA is, and it uses the base uracil rather than thymine. Both of these changes make RNA less stable. The hydroxyl group tends to participate in a reaction that breaks the phosphate backbone of the molecule, causing the polymer to break down about 10 to 100 times faster than DNA. Additionally, cytosine has a tendency to undergo a reaction called deamination, a process that turns it into uracil. Now for DNA, this is a real problem, and special molecules in your body work constantly to repair this damage. The problem with using uracil as a base is, how do you tell the deaminated cytosine from the uracil? You can't. So, how would your body know which one to fix? It wouldn't. But RNA has both of these features. one base that decomposes into another, and a hydroxyl just waiting to react with the phosphoester linkage and cleave the chain. Why do this? Why build RNA out of substandard materials? Well, the answer is simple. RNA serves as a messenger. It is designed to carry information from your DNA to a sophisticated molecular machine called the ribosome, where it's used to create proteins and enzymes that you need to survive. But what do you do with a message once it's delivered? The RNA has done its job. Now what? Well, your body does the conscientious thing. It recycles it. RNA can be broken back down into its constituent pieces and reconstructed in a new order to carry a new message. So RNA doesn't need to last you your entire life. Indeed, it is actually designed to be easier to disassemble than DNA. This way, it can be rebuilt to carry the message from a new gene and continue to drive your biochemistry. Our third and final class of biopolymers for today's discussion is polyamides. You may know them by their more biologically relevant name, proteins. Now, proteins were discovered fairly early in the development of our understanding of biochemistry. Many natural sources of fairly pure proteins are available from biological sources, such as the whites of eggs. Even in the earliest days of protein experimentation, it was noted that they had a certain nutritional value that other foodstuffs couldn't replace. And for this reason, Swedish chemist Jöns Jacob Berzelius suggested that they might be named protein, from the Greek term for first or in the lead. This name stuck. The rest is history. Proteins are the body's handymen. While DNA stores information and RNA translates and transfers information, it's our third class of biopolymer, proteins, that make up the materials in your muscles, carry oxygen to your cells for respiration, and moderate just about every chemical reaction that goes on inside your body. But just what are proteins from a chemical perspective? And how do we make sure that our bodies can produce them when they're needed? Proteins, simply put, are polymers made from monomers called amino acids. Now, this is just a few of the amino acids that your body uses to create proteins. In fact, we use about 20. Here's a small sampling of them. And they may look like they're pretty complicated, but they all follow a very simple structural theme. Now, here's a generic amino acid with the acid part at the top here, the amine functional group at the bottom, and I've designated a side chain here as R. Now, these may look very complicated, but let's break them down based on that central structure there. Notice that all of them contain that amino acid functionality. Those are all exactly the same, and they really only differ by the group that's attached to the side here, highlighted in orange. So amino acids are an interesting kind of a compound because they all have one very, very similar region and then one very, very different region. And we'll see very soon how and why both of those parts of the amino acid are critical to how they function in biological systems. For now, let's take a tour of the amino acids that your body uses to maintain its own chemistry. Here are 5 amino acids: glycine, alanine, valine, leucine, and isoleucine. And you'll notice that for each of these, the side chains are all hydrocarbon side chains. So they lack significant water solubility, and they make very good, strong dispersion force interactions with one another, but they don't necessarily interact very well in other ways. For example, think about aspartic acid and glutamic acid, which can very easily deprotonate under biological pHs to form negatively charged side chains that may have strong electrostatic interactions that those previous amino acids could not establish. Asparagine and glutamine also have what we call an amide functionality at the end of their side chain, and this gives them some interesting hydrogen bonding characteristics. 3 more, lysine, histidine, and arginine, all have the ability to take on a proton under physiological pHs, making their side chains positively charged in some cases. Yet another group of amino acids, here are serine and threonine, both of which contain OH motifs, and cysteine and methionine, both of which contain sulfur. Now, these oxygen and sulfur atoms in this particular context mean that serine and threonine are outstanding hydrogen bonding side chains, whereas cysteine and methionine are very good at forming covalent bonds to one another, meaning that they can do a process in proteins that's very similar to the vulcanization of rubber, for example, where we would get a sulfur-sulfur bridge, right, joining two proteins potentially together. We also have some other interesting amino acids at work in our bodies: phenylalanine, tryptophan, and tyrosine. Notice that these amino acids have large cyclic structures with lots of pi bonds in them. This makes them sort of large flat side chains that have the ability to stack against one another and interact strongly. So it's having these large regions of distributed pi bonds that make phenylalanine, tryptophan, and tyrosine act the way that they do. And finally, there's an interesting one, proline. This is our 20th amino acid. Notice that in proline, something's happened that hasn't happened in any of the other amino acids yet. And that is that the side chain itself has curved back around and chemically bonded to the nitrogen of the amino acid. This makes proline a very interesting amino acid from a structural perspective, because it forces the backbones of proteins, as we're about to see, into some interesting conformations that helps give them their function. So proteins are made from amino acids. But how is it that these amino acids come together chemically to create these large biopolymers that we rely on every day? Well, they come together through a condensation reaction, much like some of our synthetic polymers did previously. What I've shown here are two glycine amino acids, the simplest possible amino acids in which the side chains are actually hydrogen. Right? There's a side chain here, there's a side chain over here. They're both simply hydrogen. Now, when two amino acids come together to begin forming a protein, or a peptide as we might call it when it's very short, a condensation reaction takes place. And during that condensation reaction, water is formed. In addition to water, we have this functionality here that we call an amide bond. This may also look very familiar to you. It's the exact same type of bond that was holding together nylon 6,6 in our synthetic polymers lecture. So at its core, the backbone of proteins really isn't all that different from the nylon that you might wear, that might make up a shirt that you're wearing, or something else similar like that. Now let me add one more of these monomers, and through another condensation reaction, create an even longer chain. And the reason I've done this is I want to show you what's left over of that amino acid when it's formed a bond at both sides. So our internal glycine here now has two amide bonds holding it inside of the chain. And it's had two waters condensed and removed from it. So because it's missing a few atoms, we don't call it a glycine amino acid anymore. We now call it a glycine residue. So you'll hear me refer occasionally to amino acids that are within proteins not as amino acids themselves, but rather as residues. But why 20? DNA does just fine with only 4 monomers. Well, the reason is that many proteins are much smaller polymers than an entire strand of DNA. Many of them are less than 100 residues long, in fact. Now, having a larger library of characters increases the potential variability of smaller biopolymers, like proteins. The second reason is that proteins tend to take on some remarkably complicated shapes. The first protein structure to be solved completely was this, sperm whale myoglobin. Just about 6 decades ago, this structure was first determined by John Kendrew and Max Perutz in England. It showed not a simple uniform double helix structure like DNA, but a complex mix of various structural elements. Far from just a double helix like DNA, protein backbones can take on helical shapes, extended strands, and even turns that cause them to ball up on themselves in very specific ways. Oftentimes, the complex structures that they form are dictated by the interaction of those 20 side chains with one another. So now that we're familiar with the 20 essential amino acids and of how they come together and condense to form long chains of residues that we call proteins, let's think a little bit more about the structures that they can create when they come together. Let's start with a very, very simple chain of amino acid residues. This would be called polyglycine, because it's just one glycine residue after another in a long chain. Very simple, right? But it is indeed a protein. Now we call this sequence of amino acids a primary structure. That is to say, how they're all put together is what we would call their primary structure. So here's another example of a primary protein structure. This sequence is polyalanine. You'll notice all the side chains of my residues are the same here. They're all methyl groups, meaning that this would be polyalanine. But nature has chosen a system with many, many different amino acids available, and that gives proteins huge complexity even at this primary structural level. For example, here's one that I just threw together out of a series of different amino acids. Now you can see clearly here, I still have my polyamide backbone running through the middle of the molecule, but it's been decorated with a number of different functional groups on the sides, some of which can form bridging bonds like these sulfurs, others of which are very large and have big pi electron clouds like the tyrosine that I've got here, and so on. So all of these different side chains can interact with one another, but in order for that to happen, the chain can't be straight. It has to take on a more complex structure than simply a long chain of amino acids. Now, we call those structures secondary structures. Now, here are two very common secondary structures. One, beta sheets, shown over here, and one called alpha helices. These are still simply a long chain of amino acid residues put together using amide bonds. But you'll notice the chains aren't straight anymore necessarily. In the case of beta sheets, they are straight, but notice how they've come together and formed a network of hydrogen bonds, which creates an essentially flat structure. So when two chains come close together, they can form this interaction. Another type of secondary structure are alpha helices. And again, if you look closely here, you'll see the polyamide backbone winding through this helix, but that the angles are no longer such that it's an extended chain. Now instead it's a coiled helix. And it's packing together under the influence of the side chains, interacting with one another and changing bond angles. But we can go even further than simply looking at this secondary structure and look at something called tertiary structure. Now here's a molecule of human insulin. And its tertiary structure is a description of how its secondary structural elements pack together. We'll get them rotating here so you can see a little bit better. This is kind of hard to see because it's just a large collection of atoms. But if you look over at this cartoon representation where only the backbones are shown, you'll notice that there are several of these alpha helices and that they come close enough together that their side chains are interacting with one another. And that's holding them together in a very specific geometric arrangement that we call the protein's tertiary structure. And finally, there's a fourth order of complexity in proteins, one that we call quaternary structure. Let's look at human insulin one more time, but this time we'll look at it in one of the ways that it's stored inside of certain cells, and that is around two zinc ions, which is why you see these little gray dots in the middle here. But again, if I start this rotating and we look not so much at the large collection of atoms here, which is the entire molecule shown as balls and sticks, but rather over here at the cartoon, you'll notice that there are 6 distinct insulin molecules, but that they packed together with one another in such a way that they create this really beautiful geometric arrangement, this sort of hexagonal arrangement of proteins. And we would call this a quaternary structure because they're not covalently bonded to one another. They're simply clung together under the influences of intermolecular forces caused by the side chains making them up and their tertiary structures. At least in part, it's the fold of a protein that gives it its function. Properly folded proteins are powerful biological catalysts needed to mediate your biochemistry. Because they're so essential, improperly folded proteins can lead to debilitating illness, such as Alzheimer's disease and sickle cell anemia. Proteins generally serve one of two roles in the body. The first is as a structural material, like our skin, hair, and fingernails. And make no mistake, this is an important task, but the second role that they play can be far more critical. Proteins can also act as catalysts for chemical reactions in our bodies that keep us going. And we call these kinds of proteins enzymes. Enzymes are the worker bees of your cellular machinery, speeding up and slowing down such critical processes as oxygen transport, nutrient metabolism, and repairing that deaminated cytosine in DNA that we discussed earlier. So let's sum up today's lesson. In this lecture, we turned our attention from synthetic polymers to biopolymers. We decided to focus on the 3 most prominent classes: polysaccharides, nucleic acids, and proteins. We started with polysaccharides, where we saw that changing something as simple as the way in which a polysaccharide is branched can lead to materials as different as indigestible cellulose and your body's energy supply molecule, glycogen. We also saw how living systems can make their own semi-synthetic polymers using the example of N-acetylglucosamine in the chitin used by insects and crustaceans as a base layer for their tough exoskeletons. Then we turned our attention to nucleic acids. We discussed the features of DNA and RNA, and how each of these massive polymers is designed with a different purpose in mind. Robust, repairable DNA stores our genetic code indefinitely, while more fragile RNA is used as a transient messenger. Then we turned our attention to proteins or polyamides, and we saw how even though they're much smaller in size than DNA and RNA, they make up for that with a small library of 20 amino acid monomers. This creates extensive diversity in their structures. And we saw how they fold under the influence of interacting side chains from those 20 amino acid residues to create complex forms that dictate their function. Finally, we marveled at the incredible power of enzymes to speed up and slow down processes in the body with remarkable precision and effectiveness. And that was a perfect ending point, because our next lecture will delve into how chemists and physicians try to create molecules that will correct for the medical problems that can arise when enzymes in the body are not functioning optimally. Next up is a look at the fascinating world of drug chemistry.